I will tell you. In the cell, many organelles are present, and you know, one is a mitochondria, one is the ribosomes, the rough endoplasmic reticulum, smooth endoplasmic reticulum. We discussed those, the Golgi complex, ribosomes, mitochondria, chloroplast, and some other which I will, new organelles also, which you did not study in the 10th class, I will add in this chapter. So if you want to study one by one these organelles, you have to separate them, because they are in a combined form. They are in the combined form. They are homogenized because they are soluble almost in the cytoplasm. So what you have to do, the cell is basically a homogenized condition. Cell is in a homogenized condition. All organelles are dissolved in the cytosol, which is the liquid part of the cytoplasm. So what you will do, you have to separate these parts, and by which technique? That is the centrifugation. If you think, sir, how we were able to study nucleus, how we were able to study DNA is the hereditary material, sir, how we were able to study that chromosomes are 23 pairs in the human body, how you are able to understand the mitochondria is do respiration, how you are able to understand Golgi complex forms enzyme, sir, how we are able to know endoplasmic reticulum transport molecules from the in the form of the tubes, how we know that ribosomes form protein, sir, how we know that cell wall is composed of cellulose, sir, how you know that cell membrane is composed of lipid protein. These all things we know by studying organelles separately. Separately mean not in homogenized fashion. If they are soluble in each other, if they are together, how you will study? No, when nature made anything in the universe, the nature has given you key point how you will study. This is very important. When nature was making the cell, Allah Ta'ala was making the cell. So Allah Ta'ala knows that humans will try to understand the cells. So Allah Ta'ala give one key point for us left. What was the point? The density of each organelle, the Allah Ta'ala kept different. This is amazing. Yani all organelles have different density. Yani they have different weight. They have not different weight. They have no same weight. If they have same weight, so they will not be separated from each other. When they will not be separated from each other, you will not be able to understand. You will not be able to understand those points which you are discussing in the books. So centrifuge mean, the key point of centrifugation is that organelles are the structures inside the cell which are dissolved in the cytoplasm. You are unable to read those organelles until you will not separate them. And how you will separate them? By a machine that is called centrifugation. What is centrifugation? Just your washing machine. A rotatory thing, which is, you can see these rotators. You are watching, electric motor is there. Rotor which rotate. Due to rotation, what happened due to rotation? The heavy things will go at the bottom of the tank, and the lighter things will come up. What will happen? Each thing will be separated density wise. The organelle having a medium weight will be at the central position. Organelle having a heavier mass will be separated at the bottom, and the organelle will be at the lighter weight will come at the top of the test tube. At the top of the test tube. So by this way, these all organelles will be separated from each other. So what will happen if you want to this technique, I use the word density. Each organelles have different density. If you are talking about the density gradient, what is the density gradient? Density gradient mean density gradient configuration. You are separating the different parts of the cells due to having the different density. Each particle has a different density, and you are going to separate them by the different way. For example, if you want to separate the different organelles, so what you will do, you will put them in the centrifuge. In the centrifuge, when they will rotate in the rotatory kiln. When they will rotate, they will be separated from each other in such a way in the form of the density gradient configuration. So this is a very easy technique if you'll see, because the basic technique is that each organelle has a different density. Simple is that. When they have the different density, it is very easier to separate them by the density gradient configuration. You can see that. You can see the picture that is visible to you. Electric motor is there at the base, and rotor is there. Centrifuge tube is there. and they will rotate, and the basic thing is that the molecules which will be separated at the bottom, and the molecule will be at the top, and molecule will be at the base according to density. To mean that according to density gradient centrifugation, you are separating the particles mean organelles, due to different sizes. Some are heavier, some are lighter, and they will go into different layers in the centrifuge machine. Different layers mean, look here, this is my hand. At the top of the finger, the particles of having lighter weight will be at the top of the finger if my first layer is that. Middle, the having a medium density will be at the middle layer. With the high density will go at the bottom of the tank of the centrifuge machine. This technique is called differential centrifugation technique, and also due to how they are separated due to density gradient centrifugation. This type of centrifugation, once again I will tell, because you may be thinking, sir, what is density gradient centrifugation? Density gradient centrifugation is that you are separating the different particles or different organelles in the cell due to different sizes, due to different density, due to different masses, which will come into different layers. They will come into the different layers inside the centrifuge machine. Students, actually what will happen when you will on the rotation of the centrifuge, what will happen? The centrifuge, the particles which will be settled at the bottom, that will be called as a pellet. And similarly, the particles which will be suspended above the sediment, that will be called as a supernatant. Okay, supernatant. So these mean that particles which are settled at the bottom, they will be heavier, and the particles which are above the pellet, supernants, they will be having lighter weight. They will be having the lighter weight. In simple words, I say breathing mean taking enough oxygen into the lungs and taking out of carbon dioxide from the lungs to the outside. That is called as a breathing. So that is the breathing or external respiration. Now the main point is that plants also respire. If you are thinking that animals only respire, no, plants are also the parts of living thing. But plant respiration system is quite simple and easier one. That's why the author did not write respiration chapter name. He used gaseous exchange. So gaseous exchange take place in the humans and as well as in the plants. Now, significant pores present in the lower epidermis. You know, we discussed a lot. These lectures will be very important, students, because in the coming sessions when you will come into the classroom and these scoring sessions will start, so these all informations you have to recall, and each lecture which is upcoming lectures will depend upon the previous lecture. So my request to you that you will read my advice, you will listen these lectures as much as possible for upcoming lectures. All information are related with each other in the form of the chain. We go back to stomata. Where is the stomata situated? You know, we studied in 9th class, in the lower epidermis. Leaf has two epidermis, upper epidermis and lower epidermis. Look here, I have to show you lower and upper epidermis. Just hold on. Logi, you see upper epidermis. This is the upper epidermis and this is the lower epidermis. Both epidermis you are watching, upper epidermis and lower epidermis. Upper epidermis consists of cells, rectangular shaped cells you are watching, and lower epidermis also consists of almost similar type of the cell, but there are pores in the lower epidermis. In the lower epidermis there are pores. These pores are called stomata, singular stoma. That's why here stoma is written. Why stoma is written? Why stoma is written? Because pore is one. So between these stoma mean pore is present between bean shaped cell guard cells. So you can see the guard cells in the diagram. Guard cell, also labeled guard cell, you can see. So in the lower epidermis, what is the difference between the upper epidermis and the lower epidermis? Lower epidermis consists of the guard cell and upper epidermis do not consist of the guard cell. This is the difference. Another point of the lower epidermis. The lower epidermis consists of guard cell which is photosynthetic in the nature. Photosynthetic mean that it consists of chloroplast. Now I am telling you the cells present between upper epidermis and the lower epidermis. You can see two layers, upper epidermis and the lower epidermis. The cells between upper epidermis and the lower epidermis, this is a mesophyll cells. These are the mesophyll cells. mesophyll mesophyll cells. Why I am so sensitive in naming the mesophyll cell? These are the main cells having pigments. So if the leaf plants are doing photosynthesis, that is due to leaf. If leaf is doing photosynthesis, that is due to mesophyll cell. So that is the mesophyll cells are the main cells due to which food is produced in the photosynthesis inside the plants. These are of two type of the mesophyll cell. You can see two type of mesophyll cell. Upper cells are finger shaped, lower cells are little not finger shaped. You can see arrows also you can see, and there are some spaces in the lower epidermal cells. There are some spaces in the lower epidermal cell. These spaces are telling that there is a gaseous exchange. That's why stoma is there. Very interesting information about gaseous exchange, students, I will tell you. At daytime, the leaf is doing two processes simultaneously, mean photosynthesis and respiration. One sentence you will remind, and you will keep this point for the future. Two processes are always opposite. Which processes I am speaking now? Respiration and photosynthesis. How they are opposite? Look here. In the respiration, oxygen is used and carbon dioxide is released. In the photosynthesis, carbon dioxide is used, oxygen is released, opposite. So this is a question mark. Sir, which process take place at the daytime? Respiration will take place or photosynthesis will take place? And sir, if the respiration photosynthesis take place both processes at the daytime, which gas will enter? Because both gases are opposite. Because in photosynthesis oxygen is released, carbon dioxide is used, and in the respiration oxygen is used and carbon dioxide is released. So sir, which gas, what will be gaseous exchange through these pores? In the daytime, these pores will enter which gas? Because both opposite processes, both antagonistic processes are continuing, both opposite processes are continuing. Answer I tell you. In the daytime, students, oxygen will enter. No question. Sorry sir, if oxygen will enter in the photosynthesis, so why the carbon dioxide will not enter? Because respiration is needed in the plants 24 hours, without which they cannot remain alive. I will tell you. In the photosynthesis, carbon dioxide will be released inside leaf and that will be used for respiration. So no need to get carbon dioxide from outside. Another question arises. Sir, oxygen is not released. Why oxygen is not released? At the daytime in the leaf, oxygen is used and carbon dioxide is released. Why the carbon dioxide used and oxygen is released? Sir, why oxygen is not used in the photosynthesis? Oxygen is not used in the photosynthesis because respiration need oxygen, carbon dioxide released. In photosynthesis, oxygen released, carbon dioxide used. I am telling you again, students. In the respiration, oxygen is used and carbon dioxide released. In the photosynthesis, carbon dioxide used, oxygen released. But at the daytime, net exchange of the gas through the leaf will be two gases. Carbon dioxide will be used and oxygen will be released. No question mark. Sir, if carbon dioxide is going into leaf, how the plant is respiring? How the plant is doing respiration? Because respiration need oxygen gas. Result. Answer. Oxygen produced during photosynthesis. Oxygen produced during photosynthesis will be used in the respiration. Another point. Clear until now. I will not naming even this layer. I am interested in this layer. What is the name of this layer? This layer name is retina. retina. I will magnify. This term is called retina. Just hold on. It will come visible to you. retina. Retina. So retina is the layer. This is the retina layer. Now interesting thing is that This layer, which is the retina, this layer consists of two type of cells. Retina consists of two type of cells. Here, where they consist of this at this line, at this place. No, sorry, at this place where the fovea is written. This fovea name is incomplete. The full name is fovea centralis. Centralis, fovea centralis. Earlier I was talking about motor neuron that carry the message from the brain to the muscles and the glands. I was talking about the brain parts, forebrain, thalamus, hypothalamus. I was talking about cerebral hemisphere, which was divided into the two parts, right cerebral hemisphere, left cerebral hemisphere, and right cerebral hemisphere, which is the part of the forebrain I was talking about in the last lectures. Now I am at fovea. Full name of the fovea is fovea centralis. Veacentralis, fovea centralis. And this is the structure which consists of two type of the cells: rod shaped cell, cone shaped cell. Rod shaped cell, cone shaped cell. Cone shaped like this one, this is the cone shaped cells, and these are the rod shaped cells where they are present at this point, and this point is called as fovea centralis. This fovea centralis, which is present in the inner layer, retina, it is present in the inner layer. The name of this layer is the retina. So I was talking about earlier in the earlier lectures effectors. What are effectors? Effectors are the muscles and the glands. I was talking about the three parts of the brain: forebrain, midbrain, hindbrain. I was talking about two type of activities: voluntary activities, involuntary activities. The activities in which is under your control, mean that you can control your body. Conscious activities, those are voluntary activities, and activities which are not under your control, that are involuntary activities. And laryngopharynx, just hold on. This is the laryngopharynx. Yes, Laryngopharynx, laryngopharynx. Laryngopharynx situated above the trachea. Students, step by step I will tell you. So your system basically start from your mouth and nose, and currently you are using mask. Reason, you are just stopping air, you are just stopping virus to enter through these two holes. These two are openings which will allow virus to enter into your body, but gas also goes through this, oxygen gas also goes through these holes. So first is a mouth, and then oral cavity, the buccal cavity, and then the pharynx. Pharynx here, pharynx is the confusion. Pharynx is the confusion. The pharynx is the confusion. That is the pharynx, which is situated after the oral cavity. And the pharynx is divided into three types: the closer to the nose, that is the nasopharynx, nasopharynx, nasopharynx. Closer to oral cavity, that is the oropharynx, and closer to the larynx, that is called the laryngopharynx, laryngo pharynx, the laryngopharynx. Above the trachea, that is the laryngopharynx. So I will repeat three types. These are new terms, those you can write in your copy as well. Oropharynx, closer to pharynx, mean after oral cavity a small cavity comes, circular cavity with the muscles. It consists of muscles. It will push the food also inside digestive tube, but I am talking now respiratory system in oxygen. So this pharynx, oropharynx, look here my finger, if you can see my finger, and also in the screen you are watching these three terminologies. I am magnifying, you can see these three terms: nasopharynx, oropharynx, and laryngopharynx. Students, these informations which I will give in these lectures are very important, therefore you have to memorize, because in the upcoming scoring sessions these informations will be part of our assessment system, and you may lose the score if you will not remember these terms. Laryngopharynx, upper the trachea, upper the trachea mean your air tube, yes, which I will discuss. Laryngopharynx, above this, look at my finger, this is a top of trachea. Yes, you watching, top of trachea, glottis, glottis. Above the glottis there is a cavity, circular cavity. That is laryngopharynx. Laryngopharynx, yes. Now go back, go up. Sorry, not go back. Go up. Go up now. This is the oropharynx, and at the back side of the nose, that is the nasopharynx. So oropharynx, nasopharynx, and laryngopharynx. Now, important thing which I want to share. Glottis word I speak already. Glottis means a tube, air tube. The tube start, air tube start, and top of trachea is called the glottis. Top of trachea. This is the glottis opens into trachea, and trachea consists of mucus ciliated epithelial layer. Mucus ciliated epithelial layer. I am talking about air tube now. I am very slow because this air tube is very important, students. Air tube, trachea, trachea, EAE. Trachea consists of inner ciliated mucus epithelial layer. Top of trachea as the glottis, and above the glottis there is a lid-like structure which will control the opening and closing of the glottis, that is epiglottis, EPIGLOTTIS, epiglottis. And the students, epiglottis mean that it can close when you are eating, that your bolus will enter into your digestive tube. The epiglottis will close the glottis, and when your bolus is entering into, oxygen, when your bolus, you are not eating